Right. Good morning, everybody. Hello, my name is Vimal Patel, and welcome to our webinar today around go, no-go decisions and hindrances to them in clinical development. I'll be moderating today's discussion, and I'll introduce our panelists here in just a moment. We have some veteran panelists here with vast amount of experience. But before we bring them on, I'd like everybody to know, if you'll see at the bottom of your um, Zoom webinar page, you'll have the opportunity to ask questions or chat. Feel free as we go along to pop in a question in the Q&A. We'll try to get to many of them at the end, but there'll be some times where we may even bring them up throughout this webinar and make it interactive. So we encourage you to use that as well as the chat function for any immediate reactions to some of the things we discuss. But please interact with us. We enjoy that and we love that. But let's meet our panelists today. We've got a great couple folks here with about almost 50 years of experience in clinical development. First of all, we have Rick Graham. He's got over 25 years of experience guiding programs from IND to NDA. He's helped bring about 5 therapies to the market via global regulatory approvals, including oncology to respiratory, holding senior roles at GSK, Genentech, Onyx, Theravance Biopharma, as well as being a co-founder of TruTechnologies. He's co-founded Mirador Therapeutics and Paratrust Biologics. And Rick's helped build teams that are high performing, driving capital efficient development strategies and delivering high quality data, clinical data to make confident go, no go decisions. In addition to Rick, we've got Dr. Nancy Valente. She is a hematologist oncologist and a senior advisor at Xencor, and with nearly 3 decades of experience in drug development. She spent nearly 20 years at Genentech Roche, and she served as a global senior vice president of product development and helped guide a portfolio of over 20 hematology and oncology programs, including several transformative therapies that reach patients worldwide. And earlier in her career, she's held clinical and development leadership roles at Anosys and Coulter Pharmaceuticals and continues to serve in a variety of capacities on multiple biotech boards. So welcome, both of you.
Thanks, Vimal. Didn't realize you're going to make us sound so old, but anyway, thanks for the nice intro.
Well, it's wise. We make you sound wise, but we don't need to make you sound wise because both of you are wise.
Yeah,
So,
Nancy,
Yeah.
really nice to see you again.
Hi, everybody. Yeah.
Thank you, Nancy and Rick, for joining. So today's topic in clinical development, in your vast experience, I'm sure there have been a few consequential moments throughout your careers of go, no go decisions. And these are pretty critical moments on teams to decide, hey, do we continue to invest millions and more time and years into this asset, or do we stop? And making that right call is critical. And that's what we're going to discuss today. But before we dive into this, let's frame even what this is. What do each of you even mean? What comes to mind? What do we mean by a go, no go decision? And what are some of the factors that are critical at these moments?
Nancy, you want to go ahead?
Sure, I can start. Thanks, Rick. Yeah, so to me, the go no go decision happens throughout the life cycle of the product, from preclinical all the way to approval. These are the critical steps in the product's development, and they're unique. So there are unique decisions that are made each time, but there is probably one commonality, and that's really about the data and the quality of the data.
Yeah,
Right.
totally agree. And what I also have experienced is that these are hard decisions. There's a lot wrapped up in making those decisions. You would think that they would be easier the earlier in development the molecule is, meaning prior to going into clinical trials, but that's definitely not true. There have been researchers that have spent many, many years working on that molecule up until that point. So difficult decisions. They're not always just go, no go. Sometimes it could be pivoting to a backup molecule, but those are really kind of the general flavors of the decisions that we make.
Absolutely. Well, as we dive in, let's start with a poll question to the audience to kind of get their perspective on what did they see before we dive in on some of the biggest threats to making a sound go, no go decision. So we've got several options up there. We'll take a moment if you can select a response and see what the audience has said. I should see the results. So we see the results up on the screen. We've got 22% slow data delivery, 30% poor data quality or integrity, 35% pressure from investors, 13% emotional attachment to the asset. So as you see those results, Nancy and Rick, I mean, are those surprising? Is that what you expected? And how's that differ from your own personal experience?
Yes, not surprising. They definitely are some of the factors that go into the challenges of these pivotal decisions that are made. Yeah, all very important. Um, yeah, I think to me in particular, I think the data quality, as I mentioned before, is really essential because if you don't collect quality data, you can't answer the question you posed. So whether that's phase one, phase two, phase three, like bad information in, bad data out. So that's really important to me, right? You can't make decisions on the safety, the efficacy, the qualities of your molecule, the pharmacokinetics, the biomarkers, any piece of it, if it's not collected in a really quality, pristine manner. So that to me, that's kind of the basis for everything. The other, the other factors are there as well. People get emotionally attached to their therapies, which I think partially is kind of important, right? I'm not quite sure you do this work if you weren't emotionally involved. You wouldn't have as much um, motivation in there to like work as hard as everybody does in biotech and pharma. If you didn't feel emotional, if you didn't think the therapy that you were developing was important.
That's a good point, Nancy. Yeah, the this is my baby. There's a downside to it, but that's what you want in the people developing these therapies and giving their life's work to making sure patients get what they need during these dire times. Rick, what are your thoughts on some of the results?
Yeah, the results were interesting. I was surprised, just carrying on the emotional part, I was surprised that only 3 out of 23 said that. I agree with Nancy. And Nancy, I don't know if you remember this. We worked together at Genentech many years ago, and there was a person there that I have tremendous respect for who used to call it disciplined optimism. He wanted teams to be optimistic, but you have to be disciplined, right? So try to take the emotion out as much as possible, but be emotional in the sense that you want this project to succeed. Tracy makes a really interesting point in the chat. The data always doesn't point to a clear decision, which I think is right, but more evidence to say that we have to collect high quality data. Because if the data is noisy, which we'll touch on later, if the data is noisy, how can you make a good decision, especially when the data doesn't always tell you the clear story? I mean, so in my mind, at the end of the day, the confident decisions, which is what you're asking about here, how do we make confident go, no go decisions, comes down to high quality data and getting it as fast as you can. I mean, one of the other responses was pressure from investors. What do the investors want? They want clear proof of concept, clear data to make decisions on, but they want it fast. They want it faster than their competitors, and for good reason. So that's where things like investor pressure, competition, the emotional bias, like really get blown up, and it puts pressure on all of us developing these medicines. So,
Yeah,
um, yeah, I mean, for me, the bottom line is really that the good decisions don't really come from emotion or having more time. They come from better data.
yeah. Yeah, and you both touched on two aspects of that. One, the speed of the data, but then I think Nancy, you used the phrase that it's it's received in a pristine manner. There's high quality to it. How do you strike the balance between those two as you try to get data of trying to get as fast as you can, but as high quality as you can?
Yeah, Vimel, that's that's an issue everyone faces, probably everyone who's listening here. You want both, and you can't sacrifice, again, I'm going back to the high quality data because if you go fast and it's sloppy, 2 years later, you're not going to have anything. So you haven't saved any time. So it really is a balance in, making sure you have a good team, a good internal team. Um, you know, you're your development functions are really important, all of them, clinical operations in particular, internal, external, the partnership with the CRO if you're using one, the quality, of course, we could speak about that for another hour of the CRO. But just, and your investigators in particular, right? They have to be motivated. They have to believe in the product. They have to want to enroll. And, if those things come together, and as they start seeing some activity, that's quite important. You get more enrollment, it gets faster, they get very excited, and then everything kind of comes together. It's fun. It can be really fun and exciting.
Rick, any thoughts from your perspective on the tension?
Um, I think I would just go back to what I said before. If you follow the data and you follow the science, you'll usually be in pretty good shape. But in order to do that, you got to get rid of the noise and you got to focus on quality.
Yeah. Now, how does this change in your mind, depending on, the cycle of the development of this drug in the early phase to late stage? Does this change in any way in your perspective?
The, there are still the pivotal decisions throughout, the development of the product, but the factors that go into those decisions change. I mean, I guess if we're talking about the pressure, maybe from investors, the pressure, your own pressure to produce high quality work and bring a product you think is important forward. Um, it's the same all along the way, but let's say from preclinical to IND filing, there's a different set of factors that go into that. Like, is the toxicology, is it good? Are your preclinical models showing efficacy? Does it look like it has drug-like qualities? And then in phase 1, you're probably looking at the data a lot more often. And are you seeing the efficacy and safety you want to see, and the PK, the pharmacokinetic properties? Do the biomarkers support, it's a whole package like going in the right direction. So each time, right? And then in phase 2, it's like usually it's a randomized study, and is the experimental product, is it showing you something more than the control arm? Um, I mean, those are just some examples. Not comprehensive, but the exact factors are different. But I think what's important for all of this, and maybe um, Rick and I were just talking about this before, is having an idea of what you want to see or what you need to see. Maybe it's what you need to see to be successful, right? And what you need to see early may be slightly different than what you need to see later. Like it becomes, you gain more and more clarity as you develop the drug, but it's important to have an idea of what you would accept or reject for a safety and efficacy for each phase of development.
Mm.
Right? You want to have, before you see the data, ideally, you want to have some idea like you're expecting this percent response rate or this duration of response. You put together some parameters around safety. You don't want to see more than like X amount of this particular type of toxicity. The PK parameters, you want to have ideally like this kind of half-life, this kind of bioavailability. So
Mm.
I think both Rick and I think it's really important to have that laid out, right? You may not, like you might say this is a complete go and this is a complete rejection. Many times you fall in the middle, right? And I think that's the hardest part. Like you fall in the gray area where the drug is not a complete failure and it doesn't look like you have an 80% response rate. And maybe I'm talking about oncology in particular. Um, yeah. So
Yeah.
that's
I would just add a couple of things that, listening to Nancy talk about a priori decision making, it sounds so obvious, like, duh, why isn't everybody doing it? But surprisingly, not everybody does. I've seen a lot of different companies, and best practice is absolutely to do that. In fact, I would say best practice is for the team to come up with a priority decision making, whether that's on your research product profile or on your clinical TPP, but also go a step further and get alignment within the organization. Because the last thing you want is the team to have some data that they deliver, and then someone else in the organization like, well, no, I think we should do this.
Mm.
The only other thing I'll add on this topic, Vimo, and I'll try to take a different approach than Nancy did, which is there are a lot of external factors that influence these sorts of decisions that we have no control over, whether that's the macroeconomic conditions on a day-to-day basis, whether it's competition, there's some new data that emerges that's either positive or negative. I would rather just understand that those things are always going to happen and focus mostly on what's in your control
Mm.
and do a really good job on what's in your control.
Yeah, that's a great
Yeah,
point. Go ahead,
yeah,
Nancy.
yeah, I very much agree with Rick. Like focus on what, the data and making good decisions around that, but you're not in a vacuum, right? So you do have to consider, as you said about this, like as you're developing your molecule, years go by and the external environment changes, and just being nimble if possible with that. I mean, sometimes you can't be. You have to make a no-go decision because your product isn't competitive anymore. Or, yeah, like, right, or the money or the company's running out of money. And the product, let's say you're in CAR T-cell therapy, they're very expensive to make. So there's a lot of factors, or, there's investor pressure. I don't know, because I think most investors want the same thing as the people who work in the company for the most part. They want to see the product be successful. They just probably approach it in a different way from a different angle. But, like Rick said, of course they want it fast and they want something really stellar. And who doesn't want that, right? It's not like we want different things. It's just the reality of it is sometimes, kind of comes into play, and your plans don't work out exactly as you planned.
Yeah,
Hey,
yeah, that's great.
Vimo, Vimo, before we go on, or have you reminded the audience that they can ask questions?
Yeah, we're going to have our next poll question here shortly. But again, let me give another reminder to the audience if you join late. We're going to take questions throughout. Some of them we may save till later, but please chat and ask questions throughout, and we'll try to get to them and engage with the audience here. So, one thing I heard throughout this section is data quality, data speed is important. What are, in your mind, some of the issues, the systemic problems that sponsors need to be aware of in getting data fast with high quality? What are some of the challenges around that before we transition to the next poll?
Yes, so it's a common challenge, absolutely. And I think it gets down to, let me fast data, to me is about getting the sites to enter the data as quickly as possible. And that, they're busy, and that, you have good sites and less good sites. And there's a lot of nudging and, you know, managing of the site yourself, the company or through a CRO to help get the data in and cleaned, right? So you get it in, then you have to look at whether it makes sense. And then you have to question or query and, clean it up. So there's a lot of time that goes into it. And I think the key is kind of keeping up with it. It's easier to keep up with in some ways early phase, because you're having in phase one a lot of conversation with the investigators on a regular basis. You're hearing about basically unverified data, but you can listen to what they're telling you about what's occurring in the trial in real time and encourage them to get their data into the database. So that's the timeliness. And the quality, I mean, some a lot of the data we collect is objective, right? For the most part, it's laboratory, it's CT scans, and again, I'm in the hematology, malignant hematology and oncology area. So it's kind of objective data that doesn't require a lot of subjectivity from the investigator. There are therapeutic areas where it's a lot more subjective. It's a clinical examination, let's say of joints or things like that. So, and that's no different in, verifying and collecting quality data. I think where you lose quality data is when you don't know what you don't have, right? There's a lot of missingness and you don't know it because you weren't following the data closely enough. And it's really hard sometimes to go back and collect it, especially laboratory or safety data. And then there's, maybe Rick wants to talk about this more even, there's biomarker and other specialty lab data, which is really critical, or biopsies, that's really critical to get to the right place. And that's very different than a routine laboratory. And you have to get it from the site, sometimes under very stringent conditions, to a specialty lab so they can run the test. And I have found in my experience that that's where things really get mixed up. So PK samples get mixed up and lost. Unfortunately, biopsies get mixed up and lost. Specialty biomarker data gets mixed. You know, it's that extra when you have more handling by the site to get it where it needs to go. That's where you have more error, like in the labeling of the tubes, all those steps really lead to that, where routine chemistry and laboratory gets right to your central lab if you have one, or of course, many times the site is also running, routine lab in order to determine if the patient should be treated that day.
Nancy, great setup, and you kind of gave me a slow pitch that I'm going to take advantage of here. Wasn't planning to talk about this, but now I'm going to. And then Vimal, I'm going to come back and answer your question from my point of view on speed and quality. I mean, for those of you that don't know, my original training is as a clinical pharmacologist, so pharmacokinetics, biomarkers, and so forth. And also, if you don't know, those data, PK, biomarker data, make up a good portion of the package insert for patients. Like, how should the drug be administered? And what Nancy said is absolutely true, that a large proportion of these samples in clinical trials are lost or unusable. We know that for a fact. I know it from my own experience. Vimal, David, you guys know this from experience with customers at TruTechnologies. And I always tell people, imagine if this were a story in the New York Times or Wall Street Journal, that a patient goes into a clinical trial, and this is maybe a friend, a family member. They're taking time away from their family, from their jobs, and 25 to 30% of the biological samples we take go in the garbage. Like, that's just unacceptable. We have to do better. So that's my take on that. I want to go back to the speed question. And, some of the things I say here may or may not be provocative. I would say they probably tend to be more on the provocative side, but it's the truth. I've seen a lot in my career over 28 years. And errors happen in clinical trials way more often than we'd like to admit. When I tell people what I do, people that don't know, they're like, okay, well, they've got the impression that clinical research is really a well-oiled machine. And of course we don't make mistakes. We're developing drugs for cancer patients and, to deal with pandemics. But these errors happen, and mistakes happen throughout life, but unfortunately, sometimes these mistakes are purposeful. Fraud happens in clinical trials, and I've seen it more times than I would like to admit. Part of the problem is our payment structures. The way that we incentivize sites often is based on volume over quality, right? We typically pay sites for the number of patients they enroll, not necessarily for the quality of their work. I mean, that could look like a site cutting corners to hit enrollment numbers, or data that's inaccurately written on paper. I mean, think about it. We are writing things on paper. This is 1970s stuff when we have the technology that we do in 2025. As Nancy was saying earlier, it takes time for the sites to write things down and then later enter it into an electronic data capture system. Why later? Why not now? I mean, it's not uncommon for a busy study coordinator to capture source data on their glove or on a paper towel. Like, seriously, this is what's happening. And of course, that's going to lead to data quality issues. How can we make confident decisions on go, no go, if that's the data that we're capturing in many cases? So in my mind, the way to deal with this speed and quality issue is to use smarter systems that make capturing quality data like the way to go. That's the easy choice, not the hard one.
Well, I appreciate both of your passion on this, and that we could probably spend the entire time just on this. But I do want to kind of turn and engage the audience again. And there are some comments. We'll be looking at them. And please ask some questions throughout as well. But let's bring up the second poll question as we kind of transition to the business side of things a little bit and the impact there. And the question is, in the current climate, what's kind of the most crucial element for securing investor confidence at a go no go stage? We've got a high PTRS, a probability of technical and regulatory success, timeline to top line results, cost of clinical development, or a large potential market size. But let's give the audience a chance to kind of interact on that, and let's see what the results show there. And again, feel free to put in some questions and comments. Appreciate everybody participating.
As we're waiting, I just want to respond to a couple of the comments in chat. I wonder if Tracy's the only one on the Zoom. Are there other people on the Zoom? Tracy, thanks for your question. She's making a point about biomarker data being really important for some go no go decisions. Absolutely, right? Nancy, you were talking about, like in oncology, it's solid tumors, it's tumor size reduction. That's objective, that's different. But in a lot of cases, it's the more subjective data. I mean, biomarkers should be objective, but when there's a lot of noise in it, sometimes it can be tricky. So Tracy, your point is sample coordination is a nightmare operationally, logistically, and so forth. I would just say call Vimal Patel after this, and he can come up with a solution for you. But you're absolutely right. This is a challenge. Sylvie, I haven't read this one. Site sampling for non-standard tubes can be an issue. Lost samples, absolutely. That's what we were talking about earlier. I'm a firm believer from my own experience. Probably a third of all biological samples collected in clinical trials are lost or unusable. So anyway, thank you for your comments.
Great. Thanks for taking the poll. And the results were about 64% PTRS, and then about the low teens for everything else. What are your thoughts, Nancy and Rick? Is that surprising? Is that what you expected?
Go ahead, Nancy.
Yeah, I'm not surprised. I mean, I'm trying to figure out how to discuss high PTS. Um, you can, in my mind, I think about you can set a high bar, but then you also have to meet it, right? So I think to me that's saying you have data in phase 1 or 2. In particular, I don't know, it depends on the size of the company and who you're, what kind of investors we're talking about. But you know, you had that, to me, that means you actually produce some really stellar data. So they're excited, I guess, at any size company when you announce that your trial came out positive, and you have really important data that's going to make a difference for patients and going to change the standard of care. Yeah, they're going to be really excited. You're going to be really excited. You have high probability of success, and you have high probability of regulatory success as well, right? And maybe earlier along the way, you're seeing signals of that. So each time you see something that points you toward, like your phase 1 data is strong, it looks equal or better than other therapies out there for patients in this line of therapy. So investors are going to get excited about that because they think then when you go to phase 2, you're going to beat the control arm, right? So they're excited about that. Then you run your phase 2, you announce that you've improved upon the standard of care. They're really going to be excited about that because that randomized phase 2 leads to that phase 3, and each time you complete a phase of development, your likelihood of success goes up. So I think that's what we're meaning, and yes, of course that's important, and that's what they're going to get excited about. Let me just read.
I tried to respond to Sylvie, but
Oh, okay.
David Chung's telling me I'm not very smart, and I just sent it to the panelists. So we'll come back to that. But while we're looking at that, I just want to say that the way you asked the question led to that answer, which is what is the most crucial element for securing investor confidence? But honestly, they're all important. I interact with a lot of investors, as I'm sure Nancy does. And yeah, of course, they want a high probability of success. But if you have something that's a high probability of success that's going to cost you $1 billion and you're only going to make 500 million, that's not going to go anywhere, right? So it's really all of those things.
Mm.
Um, Competition, or let's say inherently timeline to top line results, is a really big one. I thought that would score higher, and that's largely because of how fast um, companies outside the US these days are able to bring drugs forward. So that's a really big deal. So the international drug development process sometimes is faster and it leads to more competition. All those things are important, Vimal.
Yeah. Let me ask you this, since all that those are important, what as a threat? I mean, I hear this term a lot, different people talking about noisy data. What does that mean to you too, and what impact does that have on as a contrast to having stellar data, as Nancy, as you described, and just and actually prohibiting investor confidence? But what does noisy data at this juncture mean? And can you unpack that a little bit for us, either or both of you?
So noisy data, let's probably at any phase of development is kind of the opposite of clear, pristine data. And it could mean that, it was just not high quality, so that brought in the noise. It could be that has a lot of missingness. It could be that it's a small data set. Whatever it is that decreases your confidence that you know for sure that this is going to be a product or this is going to be successful. But I think not the noisiness, because I believe we should be able to collect quality data. I think there's always inherent risk, and you're always taking a bet that it's not perfect. Like there are very few products, maybe one product I worked on in the past was Hemlibra for factor for hemophilia A. That like in phase one, it was like, oh my God, this is a product. We only need to treat a few patients. It's a rare disease. I know this is a product, but that's like one out of, one time out of, almost 30 years where it's just so absolutely clear. So it's challenging, right? So each step of the way you're trying to decide, is enough, if this is enough. If you hit it out of the ballpark and you have like 100% response, like it, that's easy. And that can be noisy, right? So if it's 100% response and you have 20 or 30% noise that brings your confidence down a little bit, you're probably still okay. So it depends on the strength and the quality of the data as well, right? If it's a really strong product, but noise means like to investors that they're not sure. Maybe that's how they describe, um, like I was describing, like a lot of times or sometimes companies will set up very clear criteria, right? So to go from phase one to phase two, I want to see this. Or to go from my phase one dose escalation to my expansion, maybe the bar is a little lower. I want to see this, right? And so you have both in safety and efficacy, let's say, an area I spend a lot of time in, parameters, and you have like I said, like this is done, like you can make a decision even early. This is a go, but a lot of times you end up in the middle, the gray area, right? And so there's kind of an art there to deciding, to looking at everything that you have, your PK data, your biomarker data, the competition, how long it's going to take to make it. What does that mean? How much is going to cost? How much money do we have on hand? When are we going to fundraise again? So all these things come together. Is this data strong enough to go to the next round of funding? What else do we need? When would that occur? I don't know if that answers.
No,
Hey, Vimal, can I try something to
sure.
add to Nancy's response? Because I couldn't agree more. Like, unfortunately, it almost always is in the gray area. I mean, I would love it if you're always on the upside of your TPP, but it's usually like, well, we didn't quite hit this, but this looks good. Let me try to paint a picture for a really simple example. Um, which is probably a more extreme example, but let's talk about a bioequivalent study for a minute, because there is no gray. It's black or it's white. So what is a bioequivalent study? That's when you're trying to show that a new drug or a new formulation behaves the same way in the body as the reference drug. So drug A, drug B, or formulation A, formulation B, are they the same or are they different? And that's determined by a statistical test, okay, bioequivalence. So in general, this is a really high bar. It's a high bar experiment to show statistical significance or not, or are they the same, are they different? And it's driven by the pharmacokinetic variability of a drug. So if you've got a drug that has a very high variability across the population, then it's going to be harder to prove that the drugs are the same, okay? So higher variability means a bigger sample size. These are big studies, they're expensive studies, and they're very important studies. For example, from a commercial perspective, if you're trying to change a formulation because the cost of goods is better, you really want that to win. You want that statistical test to show that the drugs are the same. So high PK variability can lead to a very large and expensive clinical study, and it's a risky study, even all things considered, if you design a good study. So now add to that losing data because of outdated processes, losing 25 to 30% of PK samples, and it becomes really difficult to make a confident decision. It's just, when you think about it that way, it's kind of crazy that that's what we're doing. So this is a clear case where noise can lead to a very costly and negative outcome. I also want to be respectful to Sylvie. I can't answer your question in the chat. It doesn't let me. You had asked a question, or you made a comment that some sites are using electronic medical record to transfer data directly to EDC. That's great. And I think that's great for background data on a patient because it's going to prevent errors, assuming the EMR is correct in the first place. But I don't think it solves many of the issues that we're dealing with, as Nancy's describing, and I'm describing, where we're collecting data from an ongoing trial.
Right, and keep the questions coming in comments, audience, as well. So we've talked a lot about being in the yellow, Nancy, as you described it, the ambiguity. Like, how do you approach that as a team when you're on a team? How do you approach that to be unified? How do you approach that to be, as you try to go out and raise money for the next step? And how you draw the line in a go, no go decision. But how do you approach ambiguous results, those middle ground, the gray area results?
Another challenge. Yeah, I mean, you use all, it's, let's see, I don't know if there's a clear answer here. Because ambiguous results in some settings may lead to someone saying stop. Ambiguous results in another setting may lead somebody to say, let's go on, let's get some more data, right? The data set is small, let's enroll like another 10 patients, let's make absolutely sure, right? So these are costly decisions, right? Because a lot of work, a lot of development has already gone into the product, like years of development of the actual molecule, the IND prep, everything that goes into submitting an IND, all that work, running the trial. So a lot of money and time has already been spent. So you want to make sure you don't kill a good product. At least that's how I see it. So you might lean toward like, let's enroll like another 10 patients or 20 patients if we're not absolutely sure, and really, look at that, the totality of our data, everything that we have, and make a decision then. Sometimes it's more clear, it's like ambiguous, but more clear toward the negative. And you can do that. There's a bit of an emotional investment, as we talked about earlier, to let go of something that a lot of people have invested in for a long time, invested their work, their thinking into. But it's the right thing to do, to let it go and then take that investment into a better product, take that research, take those people somewhere else. So how do you do it in a team? I mean, you work together in teams for all these products, even in small companies, and you meet together, you go over, if you're making a decision to kill the product, you want to have the cleanest data you can. So you'll take the time to make sure you have as much of the data in the company as possible, that it's cleaned up. And then you'll look at it as a group and hopefully as a team, have a recommendation, make a decision that you can present to your management team, to your CMO, to your chief development officer, to your CEO, whatever it is. It could be 5 people in the company. It could be, 80,000, but someone has to like sort through the data. People who are really close to it are probably best at determining, what this data means close to the disease state as well. That's nice. You would show it probably to your investigators as well. They're the experts. I mean, you have expertise in the company, your investigators come with a different set of expertise. And together you kind of decide like, this is good enough. It's not. And that's even early in phase one, when you're supposed to be just looking at safety, most people are looking at efficacy. So you're looking at that safety efficacy balance, that risk benefit, even early on. And the other qualities of the product. To just have the PK, the half-life, you anticipate it. As I mentioned before, is it bioavailable? Yeah. So it's challenging.
Absolutely. Yeah. I mean, and Rick, what's your thoughts from a business reality being in those situations when you're talking to investors and the like? What's that reality like?
Yeah, I mean, just to pick up on what Nancy said, I mean, there's not a like one answer for this, and it's situational. I'm generally not the smartest person in the room or on the webinar, so I like to think of simple examples. And a simple example I can think of is our job, Nancy, right, is to decrease uncertainty. Like you said, enroll more patients if it's unclear. And our job is also to try to do the right thing for patients. And so if there's a disease or a condition where there's an unmet need, there's nothing else for the patients. And the data that we have on balance suggests that the effectiveness is better than the safety profile, then I would continue developing versus stopping. But it just depends on so many things. Now, to your point about from a business point of view, I mean, look, we kind of call this the impossible triangle. You can have any two of the following three: cost, risk, and time. Okay. But you can't have all three. The holy grail is to figure out how to impact all three of those in a meaningful way. So really success comes down from a business perspective of striking a balance that's between speed and efficiency on the one side and robust and credible data on the other, like we've been talking about throughout. Investors are looking for risk profiles, so positive risk profiles of companies and their assets. And they want to see an early proof of concept. They want to see some signal in patients, ideally, that's credible. If your data is ambiguous, if it's cloudy, if it's in the gray zone, well, they might move on to the next company. So it behooves you to collect the highest quality data in the fastest time possible. You can't take forever to enroll a trial, or a competitor will eat you alive. Every day that you delay is a day that they can move ahead. So at the end of the day, the money is going to flow to clarity and probability of success. So if you have a clear data set, it can be the differentiator. You can win or you lose based on that.
Mm.
Hey, Vimo, can we answer Paul's question?
Yeah, please do.
How do you suggest to control sample collection and security? Is there an optimal mechanism? Yeah, I mean, I didn't mean for this to be a pitch on TruTechnologies or TruLab, but Paul, this company was an idea I had 12 years ago because of the egregious issues that I've seen with biological samples in clinical trials. TruLab has TruTechnologies and the product TruLab. That's the solution. Paul Vimo.
I'll be watching my phone here in a moment here. So let's move on to the third poll question as we kind of wrap up our discussion here. David, if you could pull that up. Looking forward, which innovation do you believe will have the biggest impact on improving go, no-go decisions? Is it AI, machine learning for predictive analysis? Is it live clinical dashboards? Is it risk-based quality management? And there's a lot of opportunity for advancement in this as we talked about some of the challenges, but what are some of the things that you feel like will impact?
feel like these polls are open too long. It's slow data collection.
The speed to data is not there. We're good. We got some results. About half and half for AI machine learning and live clinical dashboards. So, what are your thoughts, Nancy and Rick, on that response, both of those responses?
I agree. That's my initial thought. Yeah, I'd love to see AI and machine learning support clinical trials even more than it does now. People have been trying to put together large clinical databases for 20 years plus years. There are some challenges to that, but if machine learning and AI can support that, getting annotated clinical data in a single database such that we can use that to predict, um, to look at our early data and predict whether we're going to be successful, that, I look forward to that. We sort of do that by hand now, right? Or with minimal types of AI. I think that would be, such a game changer, and maybe someone in the audience can help correct me to say like, oh no, it's already here. Maybe Chat GPT can tell you what you need. I'm sure you can go into Chat GPT right now and ask them for your bars for phase 1 and 2 with different products. Um, yeah, so I think that's going to be really important. Um, Absolutely.
I have a different take. I mean, and this is probably just because most of my life I've swam upstream. I don't tend to follow the crowd, and I'm right more than I'm wrong, but I'm not always right. Um, there's a lot of buzz with AI and machine learning right now. Trust me, there are some really good companies out there, but I see a lot that the best they do is put it on a slide and they don't do much about it. My view on this as it relates to clinical trial conduct is let's walk before we run. Let's get rid of paper and do something better than that, and then figure out AI and machine learning. But if paper is the standard, I don't think it's a very high bar to wait for something AI related to come out.
Hmm. It's good points here. So as we're kind of coming up, as we're kind of wrap up here, and if there's any final questions you'd like to ask these two individuals, go ahead and put them in the chat. But let's one final piece before we get to our closing remarks here, just on the regulatory piece. Just a key question here, just on how do regulatory expectations, now and as they're evolving, how do they kind of impact go no go decisions in either of your minds? Um, just the changing regulatory environment, the regulatory data standards now and in the future, like how do those impact your go no go decisions?
Nancy, you want me to start on this one? Is that okay?
Yes, please.
I mean, this one's relatively straightforward for me, Vimal, because I think what we should do is remove our bias and think through the lens of a regulator, right? What's their job? Their job is to protect public safety and to make sure that every therapeutic that they approve has a favorable benefit risk, right? The benefit has to outweigh the risk. So if a sponsor is putting together a new drug application or a biologics license application, and there are parts of it that are messy because of poor quality data, then the regulator should take pause and ask themselves, is this an approvable package? Like, can I trust this? I would. I think you all would, right? So unfortunately, like here's something that's a pet peeve of mine is that the misses and the near misses that happen between a sponsor, a given sponsor and a regulator are not typically made public. Otherwise, we could all learn something from that, right? If we knew what the pitfalls and mistakes were that somebody else did, but it's kept a secret, right? People don't want to air their dirty laundry. So companies almost never share the specifics when the regulators push back, but it happens more often than people realize. So, in my opinion, credible data isn't optional. It's really the currency of trust with the health authorities.
To add on what Rick just said, which I agree with, they are looking for exactly what we are looking for. I do think there's a bit of a changing environment. I think I just read that FDA is going to start posting safety data every day. I don't know what
Mm.
that means. So we'll see. So I think we're potentially in an era of more transparency, which would be good
Mhm.
to see what's happening there. I mean, you can look at when people file the briefing packages, the FDAs and the companies, but you don't see anything along the way, right? So you don't see the little bumps. You only see the big bumps. Um, I let's see. I think the FDA's like probably under enormous pressure. Um, there's probably going to be a change. There's an evolving leadership now, and definitely people are leaving, and obviously new people are coming. So I think it's going to be a different FDA in the future. They're still doing the same job they were doing before. They appear to be doing their work in a mostly timely manner. Um, yeah, so I think it it appears to be the same. Maybe there's a little more scrutiny. Certainly, there was a recent ODAC where they brought up the issue of the lack of enrollment in the United States, and they were unhappy with that. Rick Pazder mentioned that only about 20% of patients are enrolled in the United States across the board in oncology trials. I wasn't even aware of that. And they'd like to see it go up. So that's a different challenge, what's the cause of that? I'm sure a lot of people will be thinking about that and companies will be reacting to that. It's hard, there's a lot of competition, there's a lot more products in development than there were 10 and 20 years ago. But, I don't have a crystal ball for that, but
Yeah.
that's what I see.
Well, we have come up right at the end of our time. We've c- spanned a lot of different topics and issues, and I'm sure we could keep going for another couple of hours. But I'm sure there's a number of biotechs on the call today, sitting here listening to you both. Amidst everything we've said, what would be one piece of advice you would leave for someone who is maybe in the coming months facing a critical go no decision with their asset? What one thing would you leave them with, Nancy and Rick, as we wrap up here?
I would lead with the data, like your role and your focus on the clinical data that you're collecting, like put blinders on. You can't get yourself all turned around. I don't know, maybe if you're the CEO, you get to be turned around. But if you can focus, if you're in clinical development and you can really focus on your area and just doing a really good job there, I'm repeating what Rick said, on that area, that's what's important, right? There's a lot of things that are going to come at you, especially if you're in a smaller company, and you almost have to put blinders on. Like you want to do a good job in your area, and that's why you were hired, and you want to produce the good data so decisions can be made. So you can answer the question, your hypothesis that you set out, let's say in your phase 1 trial. That would be my advice.
Yeah, pretty similar actually. I think I'll just reiterate a lot of things we talked about and try to package it into 60 seconds or less, which is follow the best practice, start with a priority decision making at the team level and get alignment throughout the organization, whatever that means at your organization. Make sure you're collecting high quality data. Do what it takes to do a good experiment and pay attention every day that you can, collecting high quality data. And then don't put off until tomorrow what you can do today. Speed really matters here, and time is of the essence. So I would sort of package it that way, Vimal. Again,
Yeah.
everything we hit on today, and really appreciate the conversation today. Nancy, always great to talk to you.
Yeah, same, Rick.
Well, thank you both for participating, sharing your thoughts, such practical and insightful comments, and I'm sure it's helpful for many of the people as they've expressed in the chat here as well. So the recording of this will be available later today. You should receive an email. But thanks everybody. Have a great day, and thanks for participating. And I appreciate again Nancy and Rick for the comments and the discussion. So have a great day, everybody.
Thank you. Bye.
You bye bye.